I call my talk a wild journey because I think in many ways that describes the experience of grad school, but also because it's more fitting in the case of my work in that I've been working with wild tomato species to look for resistance to bacterial spot disease of tomato. And this is a wild species called Solanum cheesemanii that grows in the Galapagos. And then over here we have a picture from the world's largest tomato fight. And this happens every year in Spain. Looks pretty crazy. And what I'm going to tell you about today is a different kind of tomato fight that's playing out on a molecular scale, but is arguably more significant. So just a quick outline of my talk. First I'm going to introduce bacterial spot disease and plant immunity. Then I'll talk about my work mapping a particular disease resistance locus called RZOPJ4. Then a little bit about the identification of a candidate gene for RZOPJ4 and some insights into the biochemical activities of RZOPJ4 and its cognate effector, ZOPJ4. And then about a new project to search for new sources of disease resistance. So this is bacterial spot disease of tomato. It's caused by several species of Xanthomonas. And Xanthomonas comes from the Greek for yellow unit. You can see the cells are yellow. And there are many different species of Xanthomonas that infect hundreds of different types of plants. So these are very agronomically important pathogens. And they actually made the cover of Science back in 2009. And this is a species that infects rice leaves. And you can see the bacteria actually invading into the leaf through a stomy here. And this is what bacterial spot disease looks like. So here in this field we have a resistant versus a susceptible tomato variety. And you can see how devastating it can be. And this is what some of the symptoms look like on leaves and on fruit. And it's a problem throughout the world where tomatoes are grown, especially where it's humid and where there's frequent rainfall. So in the US, this would be Florida and the Southeast, where about half of our fresh market tomatoes come from. And also in the Midwest, it's become a problem. And there's no genetic resistance currently in commercial tomato varieties, but growers try to control the disease by spraying with copper-based pesticides and the antibiotic streptomycin, but as you might imagine, the bacteria have developed resistance to both of these treatments, and they're not environmentally sustainable. So one of the things we've been interested in the Staskowitz lab is looking for sources of genetic resistance that we can put into cultivated tomatoes against this disease. And so what do I mean by genetic resistance? Here's a quick primer in plant immunity. So pathogens like bacteria, in the case of Xanthomonas, they can live on the soil, in the soil on decaying plant matter. They can survive on the leaf surface, and then they get into the leaves through stomata or wounds. And they live in the apoplast of the leaf between the plant cells. But the plant has a way to recognize these foreign invaders using what we call PRRs, or pattern recognition receptors. And these recognize conserved molecules that are common to large groups of microbes, like flagellin, shown here, or for a fungal pathogen, there are receptors that recognize chitin. And these molecules are called PAMPs, or pathogen-associated molecular patterns. So we consider this PAMP-triggered immunity. So there's an extracellular domain recognizing the flagellin, and an intracellular domain that initiates the signaling pathway, resulting in an immune response. And you can see we have a nice, healthy leaf, and the bacteria are not able to grow in the apoplast. But in the molecular arms race between the plant and the pathogen has developed this weapon called the type 3 secretion system. And this is basically like a molecular needle. It's a specialized complex of proteins that's able to deliver these effector proteins, shown as these purple stars, directly into the plant host cell. And these interfere with these immune signaling pathways initiated by PRRs, and block this immune response, and we get disease lesions. And the bacteria are able to proliferate in the apoplast. But plants have another level of defense in some hosts, and this is called effector-triggered immunity. So while PAM-triggered immunity is a rather general response, effector-triggered immunity is specific. It's classically been called gene-for-gene resistance, because any given resistance protein or R protein can only recognize one or two of these particular effectors coming from the pathogen, of which, in the case of Xanthomonas, there's usually 20 or 30 different effectors. And so this R protein, when it recognizes the effector, initiates a robust immune response and restricts the growth of the pathogen. And we can actually see this hypersensitive response. This is a rapid programmed cell death that happens and is associated with effector-triggered immunity. We can see this at the macroscopic level in the lab if we inoculate the leaves with a high concentration of bacteria, and we can see that right here. This happens within 1 or 2 days, so you're going to be seeing a lot of pictures of this, so just remember a hypersensitive response, or HR, is a cell death that's a marker of resistance. And so you might be thinking already about the next level of this molecular arms race. So say you discover your resistance gene encoding this great protein that makes the plants resistant, and you breed it into your nice high-yielding cultivated plant, and you put it out there in the field. But the pathogen has like 30 effectors, and maybe it just mutates that one effector that the resistance protein recognizes, and it's virulent again. So this is a big problem in agriculture, and so we need to think about ways of creating durable resistance. And to that end, we're interested in what are the core type 3 effectors that are conserved across many strains of the pathogen, across geographic areas and across time. And we hypothesize that these are the most important effectors for the virulence of the pathogen, and that if we have our genes recognizing these effectors, these will be the most durable in the field. And so to begin to find out what these core type 3 effectors were in our pathogen, we have been collaborating with a lab at the University of Florida in Gainesville, and they collected isolates of the species that is predominant in Florida. It's called Xanthomonas perforans, or XP, and they collected various isolates from all 5 of these fresh market tomato production areas throughout the state, and we did Illumina sequencing of 14 of these isolates and used this to predict the repertoire of type 3 effectors in each strain. And you can see the results here, well, sort of. But the main thing to notice is that we have all 5 of the production areas represented by the different colors. In white here we have 2 strains we were using in the lab that we also wanted to find out which effectors they had. And on the bottom, this is all the effectors on the left here. On the bottom you can see all these effectors are present in some isolates but not others, but all the ones in this purple box are present in all of the field isolates, and so we consider these the core effectors of XP in Florida. Although this picture may change when we have more data, so Allison Schwartz is a new graduate student in the lab, and she has now assembled 87 field isolates of XP. So as we get more sequence information, we imagine that this group of core effectors may become smaller, or we'll at least have a better picture of what are these core effectors. And the other thing to notice is these 2 effectors outlined in green here, AVRBS2 and ZOPJ4, we're particularly interested in because we have resistance genes already that recognize these. So one of these is BS2, which recognizes the effector AVRBS2, and this gene was cloned in the Staskowitz lab more than a decade ago. And these are pictures from a field trial where they compared control plants to transgenic plants expressing the BS2 gene, and you can see that under disease pressure in the field, the plants with BS2 do quite a bit better. And these actually had 2.5-fold greater yield than the control plants on average. But as I said, we can't just have one resistance gene because we already know this gene, by the way, comes from Capsicum chacarense, a wild pepper species, and it's been bred into pepper previously, so some strains cause bacterial spot disease of both pepper and tomato. But it couldn't be bred into tomato plants using traditional breeding methods because you can't cross pepper and tomato even though they're in the same family, the Solanaceae. So this approach was more effective in this case. And as I said, we already know that this resistance can begin to break down in the field. People have isolated strains that have mutations in ABRBS2 that evade recognition. So we're interested in finding more resistance genes that we can put into cultivated plants. And so when I joined the lab, I got interested in this resistance gene called RZOPJ4 that we knew from our colleagues in Florida was in this wild tomato species called Solanum pinelli. This came from Peru. And you can see that it makes small green fruits. These are about 1 centimeter in diameter, and they're not edible. And it looks quite different from cultivated tomato. And this recognizes an effector called ZOPJ4. So most of the effectors are called ZOPs. This stands for xanthomonas adder protein and reflects the fact that these are secreted out of the bacterial cell by the type 3 secretion system. So we can see here that this was an extreme example of disease symptoms where I inoculated Solanum pinelli with wild type XP, and the plants were resistant, and then in a strain where ZOPJ4 was knocked out, it was no longer recognized and disease developed. So now I'm going to talk about our work to map this disease resistance locus. So I started out using a collection of introgression lines of Solanum pinelli in a Solanum lycopersicum or cultivated tomato background. And these were developed by Yuval Eshed and Danny Zamir in the 1990s in Israel. Danny Zamir is the father of modern tomato genetics. And you'll notice in the title here that they're called Lycopersicon pinelli and Lycopersicon esculentum. So this reflects the fact, as with many organisms, the taxonomy of this group of plants has evolved as we get better DNA sequence data, and we now know that both cultivated tomato and its wild relatives are in the Solanum genus, which interestingly also includes potato and eggplant. So this collection of introgression lines has been bred so that each line has, its whole genome is like a persicum or cultivated tomato except for one segment of one chromosome where it's homozygous for Solanum pinelli sequence. And so this is chromosome 1 shown here, and you can see this IL-11 has an introgression from here to here. Each of the 12 chromosomes of tomato is covered by about 4 different introgression lines. So we have 50 in total, and these introgressions are bound by known genetic markers, which you can see up and down the chromosome. So they were developed mostly to map quantitative traits in cultivated tomato, but I was using them to interrogate the Solanum pinelli genome to see where the resistance locus was. And so what I did was I took all 50 lines and I inoculated them with a high concentration of this strain that does not contain the ZOPJ4 effector, and so I had this strain with empty vector or with the vector containing ZOPJ4. And you can see here that this one introgression line, IL-62, showed an HR that was dependent on ZOPJ4. So that showed us that the locus was here on chromosome 6. Sorry, it's a bit fuzzy. And then there was a sub-introgression line called 622, and that also showed the HR, so that narrowed it down quite a bit. And then 6.1, which overlaps with those, was susceptible. And so that showed us the locus was here, and this is around 8 centimorgans. So these introgression lines were actually not good as resistant parents of a mapping population because they had this linkage drag problem. So you can see here that this is the cultivated tomato parent. These plants are all about the same age. It looks nice and healthy, but then the 6-2 and 6-2-2 introgression lines have an ontogenious necrosis, so as the leaves get older, they tend to shrivel up and die, and the plants don't make a lot of fruit or seed, they don't germinate well, so they were kind of a mess. So we didn't want to use these for the mapping study. And this problem of linkage drag is common in plant breeding, where you have an interspecific hybrid between a cultivated plant and a wild species, where the trait you want, the disease resistance, is literally dragging along with it a lot of other genes from the wild species that are conferring these undesirable traits. But fortunately, our colleague Bob Stahl in Florida had been working on breeding for this trait for a while, and so he had a 7th backcross line, which we had to call OC7 for outcross 7, but it's basically a backcross. And this had gotten rid of a lot of that linkage drag phenotype. So we were able to use this as the resistant parent of a mapping population. And you can see here that it also shows the HR and disease resistance that's dependent on ZOPJ4. And we could quantify this resistance by inoculating with 3 different strains of XP. So this one is wild type, the white is a ZOPJ4 knockout, and then the dark gray is a complemented knockout. And we inoculated susceptible F8000 tomato line, the wild species Solanum pinelli, and the 7th backcross line. And you can see when you start the growth assay, they're all about the same. And then after 9 days of growth, you can see that in the susceptible plant, all the strains grow to about the same level, maybe slightly less than the ZOPJ4 knockout, since it's contributing somewhat to the virulence of the pathogen. And then in Solanum pinelli and our 7th backcross resistance line, we see that there's a disease resistance to lower bacterial growth, and it's dependent on ZOPJ4. And since this is a log scale, these are actually about 5 to 10-fold differences in the level of bacteria. And so this was our approach for map-based cloning. This is a classical plant genetics technique that I had the privilege of learning in grad school. It's actually pretty cool when it works. So here, these are not representing whole chromosomes, but you can think of them as the region of chromosome 6 where we've already narrowed down the resistance locus. And here we have our F8000 susceptible tomato genotype in white and the resistant genotype in black. And we cross these 2 parents. We get a heterozygous F1 plant. We allow that to self-fertilize. And then in the F2 generation, if you remember your Mendelian genetics, our resistance segregates as a single semi-dominant locus, so we get a ratio of 1 to 2 to 1 of these genotypes. And what I mean by semi-dominant is that these plants that were homozygous for Lycopersicum were susceptible, no HR. These homozygous resistant had HR and showed the disease resistance, but the heterozygotes were somewhere in between. So you probably can't see this, but they often showed a weak HR and kind of an in-between level of disease resistance. Sometimes they were resistant. So we call this semi-dominant, and this is somewhat common with disease resistance genes in plants. But importantly, in the F2 generation, we can take advantage of the natural homologous recombination that occurs during meiosis in the F1, and we have a rare, a small percentage of individuals in the F2 population. So in our case, we had 3% of the 910 F2s that we genotyped had a recombination event on one copy of the chromosome across this region where we knew our ZOPJ4 was. So based on the genotypes and phenotypes of those recombinant plants, we could narrow down where the disease resistance locus was. And the way that we genotype the plants was using these molecular markers. So it's an old-fashioned technique, mapping, but we had some somewhat modern data and tools that we could use to do this. So I made a lot of use of a website called the Salt Genomics Network out of Cornell for solanaceous plants, and the tomato genome was in progress while I was working on this project. So that made it kind of interesting, and the data that we had to develop these markers kept getting better and better. And the genome was finally published last year in Nature. So one of my markers was a simple sequence repeat, or SSR marker, and this just spans a region of TA repeats in the genome, and it's longer in Lycopersicum than Pinelli, so if you PCR out this region, you can see that you get a bigger band in Lycopersicum than Pinelli, and importantly, it's co-dominant, so you can see the heterozygote has both bands. And then most of the markers were cleaved amplified polymorphic sequence, or CAPS markers, so these take advantage of a single nucleotide difference between Pinelli and Lycopersicum, where in this case there's a restriction site for the enzyme NC2 in Pinelli, and you can see that this PCR product in Pinelli gets cut, and in Lycopersicum it doesn't, and it's also co-dominant. So we ended up with 14 molecular markers across this region that we used to genotype the F2 plants. And this is showing the genotypes and phenotypes of the most informative recombinants. And so this is how we delineated the locus. So these two were susceptible, but they had Pinelli sequence here, so that showed us the gene must be this direction. That gives us that boundary. And then this plant was resistant, and it had Pinelli DNA through here, so the gene must be this way. So that gives us this, and so we know that the gene is somewhere in this 190 kb. And we published this work last year in Theoretical and Applied Genetics. And so the next step after that was to identify a candidate gene, or several candidate genes, to begin with. And before I talk about that, I need to go back and tell you what kind of genes I was looking for. So remember our effector-triggered immunity, so we're looking for something that encodes one of these R proteins that could be recognizing an effector from the pathogen. And there are 5 classes of plant disease resistance proteins that have been characterized. The vast majority of them are what we in the field affectionately like to call nibblers. So the NB stands for nucleotide binding, that's this central conserved domain. And the LRR is a leucine rich repeat domain that's more variable and seems to confer the recognition specificity of these proteins. And then the N-terminal domains are also variable. And then another type are these receptor-like proteins, exemplified by the CF proteins, which are fungal resistance proteins from tomato as well. And these have an extracellular LRR. Then we also have intracellular kinases, such as PTO, which is a tomato bacterial spec resistance protein. And then we have these receptor-like kinases, which are kind of a hybrid of those two. And then RPW8 is a unique one that sticks in the membrane and then has an intracellular coiled domain. So we were looking for any genes that would encode something that looked like those 5 things. And so if we look more closely at our 190 kb, on the top we have Solanum lycopersicum, cultivated tomato, and then our resistant wild species on the bottom. And I was fortunate enough that a group in Germany was also working on sequencing Solanum pinelli, so I had this nice big scaffold. By the time we had narrowed the locus down to 190 kb, I had this sequence, which was wonderful. And you can see here the purple boxes are our genetic markers, and then there were around 30 genes in this region in both species, and the only things that looked like disease resistance proteins looked like one of these intracellular kinases. They're not very big proteins, they're around 300 to 400 amino acids, and you can see here that this one we're calling 06G, 060680, that's its official tomato annotation number. And this one had a close ortholog in Solanum pinelii, and then there were 3 other kinase genes in Lycopersicum, and then 2 in pinelii. And so we have 3 candidate serine threonine protein kinase genes in pinelii to look at. And the way that I started working with these candidate genes was using this Agrobacterium-mediated transient expression assay. So Agrobacterium is another plant pathogen that acts like a natural genetic engineer. As many of you probably know, it introduces DNA into the plant where it can be expressed and produce protein. And the host that we use here is Nicotiana benthamiana, so this is a wild tobacco species that's very amenable to these types of assays. And this is an established technique for studying the interactions between effectors and their cognate resistance proteins. We literally just squirt the bacteria into different spots on the leaf, and it's nice because you can do several spots on one leaf to compare the responses. And this is what I used as a control. So I mentioned before the effector in our protein ABR-BS2 and BS2. And we usually have these under the control of the 35S promoter, so they're highly expressed in the plant. So we inoculate with Agrobacterium carrying empty vector, and then ABR-BS2 by itself, BS2 by itself, and then we only get this hypersensitive response when we express both proteins together. So I tried this with the 3 kinases and this apoptotic vector, and hopefully you can see here that only kinase 1, not 0680 or kinase 2, produces this HR when co-expressed with SOPJ4. And we always want to check whether our protein is produced. All the proteins are tagged with HA epitope. And so on this Western blot, you can see that 0680 was expressed as well as kinase 1. Kinase 2 was unfortunately not expressed. This was the only one of the 3 that had predicted introns, so it's possible it wasn't spliced properly in Nicotiana. Haven't been able to follow up on that, but in any case, this was a very strong indication that kinase 1 was the RSOPJ4 resistance gene. But to really know if it's functioning in disease resistance, we need to put it into a tomato plant and see if that's the gene that's conferring the resistance. And I sent constructs off to do this back in the fall to the University of Nebraska, to their core facility. And then in January, I got back my little plantlets in little cups. And exactly 3 months later to the day, so this was just 2 days ago, I got the first ripe fruits from some of the primary transgenics. So I tried a little bit, but it's very hard to assay the resistance phenotype in these primary transgenics. They're hemizygous and they're not quite normal, so we have to wait for the next generation. So 1 out of 4 of these seeds is going to be homozygous for the transgene, and so stay tuned. So we're waiting for that last big piece of evidence. But in the meantime, we can look at similarities between both the effector and the putative R protein and known resistance proteins and effectors to think a little bit about how they might be functioning. And so I'm going to talk again about PTO. So PTO is a very famous disease resistance protein. It was the first of this type of gene-for-gene resistance proteins to be cloned from tomato. It confers resistance to bacterial speck disease caused by Pseudomonas syringae. And we know quite a bit about the way this effector works. So it recognizes ABR-PTO that's secreted by the pathogen, and this effector interferes with the kinase domains of these pattern recognition receptors, including FLS2 that recognizes flagellin, to block this PAMP-triggered immunity. But then PTO actually evolved to mimic the kinase domains of these pattern recognition receptors, and then it interacts with a more classic R protein, a nibbler called PRF, to initiate this effector-triggered immunity for cell death. And it seems that ABR-PTO is actually inhibiting the kinase activity of PTO, and we know that there's this conserved threonine residue here in the activation loop of the kinase, so this is a alignment of RSOPJ4 and then two of the other kinases from this locus, and you can see this threonine residue is conserved, and there's evidence from PTO that this residue is required for autophosphorylation and also for its direct interaction with ABR-PTO and for signaling the immune response. So I thought if I mutated this residue to alanine, it might also be required for the RSOPJ4 recognition, and I saw that this was the case. So if we co-express wild-type RSOPJ4 with ZOPJ4, we get the cell death response, but not with the T233A mutant, and we can see here that the protein is still expressed. And then what about the effector? So ZOPJ4 is part of a quite interesting family of type 3 effectors that are shared among both plant and animal pathogens. And YAPJ is the founding member of this family. It comes from Yersinia pestis, which caused the Black Death, that, as you probably know, wiped out about a third of Europe's population in the Middle Ages. So this is one of the effectors that allows pathogens to really wreak havoc on their hosts. And we can see here a phylogeny of the YAPJ family. So YAPJ is down here along with some other effectors from animal pathogens, and then our effector, which used to be called ABRXV4, it's now called ZOPJ4. That's up here. Its closest homolog is from Ralstonia, which is another plant pathogen. And the function of this effector family was somewhat of a mystery for a while because based on sequence, it seemed that these were similar to cysteine proteases, but people couldn't identify any host targets that were proteolized by the effectors, and they seem to have very weak protease activity in vitro. And then this paper came out in 2006 from Kim Ort's lab, who had worked on YAPJ for a while, and they found that YAPJ was actually acting as an acetyltransferase, and it was acetylating kinases in mammal cells that were important for immune signaling, thus blocking their phosphorylation and blocking immune responses. And then a similar pattern was found for HOPZ1A, which is here in the phylogeny. So this is a Pseudomonas syringae effector, and it was found to acetylate tubulin, which is a quite interesting virulence tactic. So it interferes with the plant cell cytoskeleton, thus interfering with secretion and with immune responses. And so what about ZOPJ4? So we have a little bit of evidence that it might be functioning as an acetyltransferase, and this comes from our collaborator Mary Beth Mudgett at Stanford. So she took in vitro purified proteins and incubated them with carbon-14 labeled acetyl-CoA, and you can see whether the protein gets radioactively labeled. So HOPZ1A served as a positive control here, and then here's ZOPJ4, so we can see it's indeed autoacetylated. So that's some evidence that it may be functioning as an acetyltransferase. And another thing that we know about the YAPJ family is they all have this conserved catalytic triad of histidine, glutamic acid, and cysteine. And the cysteine seems to be the most important residue for the catalytic activity. It's thought to be the one that first makes this covalent bond with the acetyl-CoA. And so if we mutate this residue to alanine, we can see in the transient assay that while the wild-type ZOPJ4 is recognized by our ZOPJ4, the C219A mutant is not, and the protein is still made. So we have some ideas about how these proteins might be functioning. And so we can think about models of effector recognition based on other are proteins and effectors that have been studied. So we know some instances of a direct recognition where in green we have the effector protein, and then this is a classical MBS-LRR resistance protein, and we know that in some cases they directly interact. But what seems to be more common is this guard or decoy model, where the effector is modifying a host protein that serves as its virulence target in the case of the guard model, and then that modification is recognized by a nibbler. Or in some cases, like PTO, this protein has evolved to mimic proteins that are actually involved in immunity, and its purpose is for the nibbler protein to recognize that modification by the effector and initiate immunity. And then there's also a bait model where another host protein is required to facilitate this interaction. So it's kind of tempting to speculate that, based on what we know about YAPJ, acetylating kinases in animal cells, maybe ZOPJ4 is actually having a similar activity where it's acetylating kinases that are important for immunity, and maybe our ZOPJ4 is either a virulence target or it could be a decoy that evolved to mimic the real virulence targets of ZOPJ4. And there may be a nibbler protein involved that recognizes this activity, but at this point we can only speculate. And you might be wondering, then what happens after this initial recognition of the effector by the resistance protein? How does that result in an immune response? And that's quite an active area of research, as you can see. So this is from a recent review on PTO, and there are many genes that have been found to be required for this immune response, and they involve a lot of different cellular processes that go on in the chloroplast and peroxisomes, and of course gene transcription changes in the nucleus and effects on secretion, and intriguingly a kinase cascade. So kinase signaling is important for immunity, which might tie into our recognition story, but the main message is that it's quite complex. And so now I'm going to switch gears a little bit and talk about a project to find new sources of disease resistance, because as we already saw, if we have just one R gene recognizing one effector, the pathogen mutates that effector, it still causes disease, you have a problem again. So what we hope is to be able to stack several resistance genes that recognize conserved pathogen effectors to create more durable resistance. So we have 2, 3 would be even better, more would be even better. So to find more sources of resistance to bacterial spot, I enlisted the help of Josh Chu, an undergraduate who's been working with me in the lab. And we took a collection of wild tomato relatives from the Tomato Genetics Resource Center at UC Davis, and he did dip inoculations on these. So this is a little bit more similar to natural conditions than directly injecting the plant. So you have a suspension of bacteria, you add surfactant so that it can coat the leaf surface, and then you keep the plants under humid conditions. And so this allows the plants, the bacteria, to stay on the leaf surface and get in through the stomata and cause these nice greasy disease lesions after about 7 to 10 days. And so the control here was FL8000 susceptible tomato, and then we had plants with the BS2 and our ZOPJ4 resistance genes. And you can see here that these are just 3 examples of the many susceptible wild accessions that Josh found. I think he's screened around 100 or so now. And there are 9 candidate-resistant accessions. So these show no disease symptoms under those conditions. And he also quantified the bacterial growth in some cases. So this was after 7 days. You just grind up the leaves and plate out dilutions of the bacteria. So our susceptible control has quite a high load of bacterial growth, while the BS2 plant does not. And then three of our resistant candidates were somewhere in between. And again, this is a log scale, so this is our most promising candidate, Solanum pimpinellifolium 0722, and this is almost 100-fold less bacteria there. And then another thing he did with this accession was look to see if we could find out which effector was being recognized. So with an HR assay, so this is again a high inoculum of bacteria after a couple of days to see if we get the HR. And you can see the wild type XP gives the HR, as well as all 5 of these effector knockouts, so that tells us that it's not one of those 5 effectors being recognized, which is good in a way because 2 of them are ZapJ4 and ABRBS2, and we already have resistance genes that recognize those. But if you look at this HARC-V knockout, the HARC-V is a conserved component of the type 3 secretion system, so this strain doesn't secrete any effectors, and that doesn't give HR, so this at least suggests that there's some type 3 effector being secreted by the pathogen that's recognized in this accession. And so where did these wild species come from? They came, well, for us they came from UC Davis, but they have been collected over many years throughout the world, and mainly from this part of South America. So this is Peru, Ecuador, Peru, and Bolivia. It's the foothills of the Andes, which is the center of origin of tomato. And so that's where we find great genetic diversity and its wild relatives and thus disease resistance. And you can see here that actually 6 of our 9 were Solanum lycophyrsivum, or cultivated tomatoes, so there might be more diversity there than we realized. And 2 of those came from Brazil, and 1, interestingly, came from Sri Lanka. And of course all this needs to be followed up, but we certainly have some promising candidates, and the next step now is to cross these back to cultivated tomato to begin the process of mapping these new resistance genes, which hopefully will be accelerated now through the use of next generation sequencing. And so to summarize, I've told you that Xanthomonas perforans causes bacterial spot disease of tomato by using a type 3 secretion system to subvert the plant immune system. And the conserved type 3 effector, ZapJ4, is recognized by the R-ZapJ4 resistance gene from Solanum pinnellii. We used molecular mapping to define the R-ZapJ4 locus to 190 kb on chromosome 6. And then transient co-expression to evaluate these 3 candidate kinases and reveal a promising R-ZapJ4 candidate. We looked at some conserved residues in both proteins to find out a little bit about the mechanism of recognition. And we're still waiting for the homozygous transgenic tomato plants to really demonstrate that this is the disease resistance gene. But in the meantime, we identified 9 potential new sources of disease resistance. And so I have a lot of thank yous. Ryan's been great to work with. He's always very enthusiastic about the science, but he also always makes sure that your experiments are logical and that you have the right controls. And Doug has been great with his technical expertise, as well as his general good attitude. And the undergrads I've had the pleasure of working with, Josh, Richard, and Lily, have been wonderful. Everyone in the lab has helped me at some point or another. And I have to thank my qualifying exam and dissertation committee members, particularly Steve, for allowing us to use his humidity chambers to do all that screening of the wild accessions. And our collaborators at the University of Florida, UC Davis, Stanford, Max Planck in Germany, the department staff, and especially the greenhouse staff for keeping up all our plants and Barb for having everything run smoothly. The core facility at the University of Nebraska for the transgenics, and NSF and Two Blades Foundation for the funding. And here's the lab. These are just such excellent people. It's been so wonderful to work with all of them. And Brian always tried hard to make sure that we worked hard and played hard. So he invited us to his place in Tahoe. We went hiking. I wasn't there for this wine tasting. I've been there for others. We have a good time in the stats lab. And I had to try to embarrass Brian a little bit. So this was from a couple of Halloweens ago when everyone dressed as Brian. He wasn't there.
Kathleen Ryan's still going to be paid back one day.
I know it's coming, I know.
You know when it's not coming.
My Wondergrads, I took that term from you, Jennifer Lewis, thank you. Lily and Josh, it's just been so much fun to work with both of them. They're both here today. And to watch them learn and help them learn, and I learned a lot from them too. My classmates, everyone is showing the so-called naked poster session picture. And I also decided to show the other 5 people who weren't in that picture. I've just made great friends in my class, and I have to especially thank Renu Kumar for living with me the first 2 years of grad school and being such a great friend. I love these guys. And so you can see based on this evidence that I've had a long-standing interest in plant biology. Years to this day. And I have to really thank my parents for always encouraging my curiosity and my interest in biology. They couldn't be here today, but they're coming in a few weeks. And I was really proud of my dad because while I was in grad school a few summers ago, he actually rode his bike across the country from west to east. I don't have any pictures from the west end, but I was there at both ends, which was awesome. And so here's the 3 of us when he finished. This is on a beach in New Hampshire, and triumphantly holding his bike over his head. So that was pretty great. And then I have to thank my husband, Kevin, who moved from Boston to California to be here with me and convinced me to get a cat. Who we named Martin and actually turned out to be pretty great. Good source of comic relief and also just a general sweetheart. He turned me into a cat lover, which I never thought would happen. And we got married in 2011, and Kevin's just been a great hiking and travel buddy and just all around wonderful. So, thanks. I think that's all I have to say.